one of the goals of SSCR is promoting research in general. So can you give us a little description of what your day-to-day is like and how the lab functions? So I know you said there were 6 people in the lab. Would you say there's a lot of independent work, collaborative work? And then I guess a little bit about the pandemic, so how that's affected your day-to-day operations.
Yeah, those are good questions. So the life of a scientist is basically what you want me to describe.
Yeah.
So my work, especially the work that we do working with stem cells, human stem cells, it does require a team of people to work collaboratively. While they each have their own independent research project, they work together in the sense that they help one another out with their cell culture work. Working with human iPS cells require daily maintenance. And so if you work independently, you can imagine being here 7 days a week, every day until your project is done. So for them to work together and have that harmony is important because then they can help one another out. That is the nature of the work that we do. Of course, not all labs are like this. You work with cell lines, they're much more easier to deal with. But working with iPS cells, human stem cells especially, does require people to work a little bit harder. But because we have such a collaborative niche, we have a good team that works very well together and trust one another, they're able to distribute that kind of responsibility amongst the 6 of them. So yeah, so basically they come in, they meet with me once a week or sometimes more if they need to. I have an open door policy. They can come knock on my door for help anytime. So they meet with me usually earlier in the week, Monday or Tuesday. And then we go over the plans for the week, what they will do. At the same time, they also give me up-to-date updates on what they've done the previous week. And if there were any problems, they let me know and we try to problem solve together, so that we can, fix the problem. So we meet basically an hour once a week, one-on-one. They go off, they do their proposed plan that they had planned out to do for the week. We meet again as a team once a week on a separate day for lab meeting, and there one person presents a holistic view of their project. It gives them a good practice in terms of publicly speaking about their work. So, and that I find is very useful because you get the entire lab contributing to the lab meeting, right? Providing input and feedback and even suggestions, especially to their colleague. And then we also meet once a week to discuss what other people have published in the field. So we call that journal club. So I actually just came back from a journal club. So one person will present a paper that they thought was exciting and wanted to really discuss. And then we all talk about it together and give our input and suggestions sometimes of how it could be done better, or if we were to do it, how would we do it, etc. Or if it's a fantastic paper, it's like, oh, those are great ideas, maybe we should adopt them, kind of thing. So that's usually the gist of what we do and the day-to-day life. I don't think it's like any other job other than you work with exciting cells.
That's good to hear. And then the second part of the question was like, because of COVID, have you guys decreased the amount of times you're coming into the lab or separating maybe like the lab parts into independent?
Yeah, absolutely. So this affects everyone, not just our lab. So at Sick Kids right now, we're in phase 3. So this building can only have 60% occupancy at one time. And because of that, we do have a schedule where they have to sign up when they will be coming to the lab. We try to make it fair so that everyone has their chance to be in the lab and to get their work done. So yeah, we're working at 60% capacity, just because that's the rule right now. And of course, with the rising numbers of COVID cases, we might have to dial back to one of the earlier phases, which is not what we would want. Having said that, there is a small team in my lab that gets to come in at 100%, simply because we have COVID funding, so funding to do a COVID project with our stem cells. So I mentioned that we're using our stem cell lung models to start a respiratory infection. So COVID is the one that we're looking at. And
Mm.
so we're trying to see how the virus is affecting the epithelial cells, the lung epithelial cells, and see if we can learn anything from that to find a target to treat COVID patients with. So,
Is there anything that you found so far that you're only
not yet. We're
sharing?
still in the process of ramping up. So because there was a shutdown earlier on in the year, no one was able to come to work, and because of that, all our cultures had to be frozen down. So ramping up has been a process.
Yeah.
Um, Also because if they don't allow many people back, it makes it very hard to ramp up. And so right now we're almost there. We're almost at the capacity where we can now take ourselves into the CL3 facility at U of T. So it is a designated facility to work with that particular virus. And so once we have those numbers of cells that we need to do the live virus infection, we can then start looking into some interesting, hopefully some interesting data.